All right, so we're picking up now with our third section on the green plants, and from here on out, we're going to be dealing with the tracheophyte plants. So, tracheophytes, again, that just means that these plants have, at a minimum, tracheids, and some of them also have not only tracheids, but also vessels. So, what we're saying there is they have vascular tissue, which means they can get bigger, they have more structural support, they have better capacity to transport water. And so these are really the plants that most of us, I think, are most familiar with because they're things like oak trees and pine trees and maple trees, and they're really obvious things that most people think of when they think of what a tree actually is. Um, so we're going to start off here, and we're going to look at the three kind of main groups of tracheophytes. The first group being the lycophytes, which are the most primitive of the tracheophyte plants. They're the first ones that actually kind of came up with or invented or evolved the vascular tissue. We'll then take a look at the pterophytes, which are the true ferns, the whisk ferns, and the horsetails. And then we'll take a look at the seed plants, which are gymnosperms and angiosperms. All right, so our three big groups there of tracheophytes. Now, as this progression took place, as this evolutionary trajectory sort of occurred with the development of bigger plants that have vascular tissue, the other big trend that occurred is that gametophytes got smaller and smaller and smaller, while sporophytes got bigger and bigger and bigger. So by the time we get to the actual seed plants, again, maple trees and oak trees and pine trees and so forth, their gametophytes are actually microscopic, and the sporophyte is the entire rest of the plant. The thing that you see when you look at a tree from a distance is actually the sporophyte. To see the gametophyte, you'd actually have to have a microscope, and of course, it would only occur during certain times of the year when cones are fertile or when flowers are present or those kinds of things. So gametophytes and the associated gametangent get smaller, sporophytes get bigger. That's kind of the progression that we see in this process. So the earliest of tracheophyte plants had stems, and so again, remember to be a stem, it has to actually have vascular tissue, has to have xylem and phloem, but they didn't have any true roots, so they still had rhizoids, a lot like what, say, a moss would have. And they also didn't have true leaves. So they had root like structures, they had leaf like structures, but because those structures didn't have vascular tissue, they were not technically, officially, scientifically roots or leaves. Um, so the lack of roots meant that the early tracheophyte plants and even modern day sort of primitive tracheophyte plants didn't really have the ability to get very large, because A, they didn't have a very firm base, they didn't have a really good anchor to the ground, and B, they didn't have a whole lot of capacity to absorb water from the ground. So they were still relatively small and still are today. Eventually, roots developed, and so as we've seen before, roots are involved in not only getting minerals and nutrients and water from the ground, but also support. It's a good solid base for the plant to be able to stand up to have these good roots. Lycophytes, even modern day ones, club mosses, still today do not have true roots. So the development of actual roots occurred after the lycophytes split off from the rest of the plants that gave rise to things like ferns and whisk ferns and horsetails and seed plants. Lycophytes split off. They didn't have roots at that time. They still don't, and they still do okay. They're not a huge group of plants, but they still, for those that are out there, do okay in the environment. Leaves developed as well. So leaves, primarily are involved in various aspects of photosynthesis, whether it's gas exchange, whether it's the actual photosynthetic process itself. Also, water loss occurs through the leaves. But again, to be a leaf, it has to have true vascular tissue. If it doesn't, then we would just call it a leaf-like structure. So there actually are two different kinds of leaves out there. Euphylls are true leaves, and they're things that ferns have and seed plants have, and most of us are most familiar with euphylls that have multiple veins, relatively large kinds of structures. Lycophylls are the leaves that developed, or leaf-like structures that developed in the club mosses, and they're a lot smaller. And really, they only have one vein per leaf, so they're actually very, very tiny, single-veined leaves. So the development of lycophylls probably looks something like this. Originally, lycophylls plants had a stem with a vein. So you can imagine if there was some sort of a mutation that occurred that caused these plants to give rise to branches. Eventually, these branches would be better supported if the vein continued off into the side here, like we see over here. And so now what we have is an offshoot of this main stem that has a vein in it. So therefore, at that point in time, that offshoot technically could be considered a leaf. And eventually they became a little bit flatter and broader and better able to pick up sunlight and CO2 and so forth, get rid of oxygen as well. But that's essentially what a lycophyllous leaf would look like. Euphylls, on the other hand, have leaves with multiple branching veins. So, and again, this is, more like what we'd imagine today with something like a maple leaf or an oak leaf, or even pines and things like that have these as well, where the leaves have multiple branching veins in them, as opposed to just one vein like we see in the lycophylls up top there. All right, so with the tracheophyte plants evolving, um, around the same time as animal evolution really took off as well. It was about 400 million years between the time when vascular tissue first showed up in stems and vascular tissue showed up in leaves. All right, so if we jump back a second here, this progression from something like even this to this was about 400 million years. So not a very short period of time at all. It's actually about a half a billion years. So one of the reasons why this new type of leaf-like structure was favored was the fact that around this time there was much less carbon dioxide in the atmosphere than what there had been previously. So sometimes what people don't understand is that carbon dioxide fluctuations occur naturally all the time. Now, the CO2 that we're putting into the atmosphere as humans is a bit of a different story because A, it's caused by a species, us, which has never really occurred before in the history of life on the planet, and B, because this happened so quickly. But carbon dioxide fluctuations have occurred throughout Earth's history. There have been times when CO2 levels were relatively high, there have been times when CO2 levels were relatively low, and during those times when CO2 levels were low, for plants at least, having flat, broad structures able to absorb CO2, get the most out of the little bit that's in the atmosphere, was actually a benefit and allowed them to get more carbon dioxide to maintain that photosynthetic process. The other aspect of leaves is that by having stomata, which we've seen before in this chapter, we also saw it back in Principles 1, is that stomata allow for evaporation of water. And so by having evaporative cooling of the leaves, bit of a double-edged sword because yeah, it lets the leaves cool down, but it also allows for water loss, which if it's, too extreme, that can actually be a bit of a disadvantage. So water loss up to a point is good because it helps cool the leaves, but beyond a certain point, it actually becomes counterproductive and causes plants to desiccate or dehydrate over time. Now, the other big leap forward, in addition to vascular tissue in stems and leaves and roots, was the development of seeds. Now, one time this has been a long time ago, I read a botany book that talked about a seed as being a baby plant in a box with its lunch, which always kind of stuck with me. It's it's simplistic, it's kind of cutesy, but I actually like it because it really kind of sums up what a seed is for a plant. It is an embryo. It does have a wall around it, which is protective, and it does have food inside of it. So a baby plant in a box with a lunch is really not all that far off. So it's a way for seeds to get through the tough times. If seeds are produced in an environment that's kind of harsh or inhospitable, they can be dormant for sometimes thousands of years. People have actually found seeds buried in Egyptian tombs that are, 4,000 years old and they're still viable. They've taken them to, a lab type situation, they planted them and they actually grow after 4,000 years. So seeds are a really good way for plants to make it through the tough times and only start to grow when conditions improve. Seeds can also be dispersed very widely depending on what kinds of seeds we're dealing with, especially if fruits are in the equation as well. There's a lot of possibility for dispersal to take place once seeds are part of the equation for plants as well. Now, the lycophyte plants, which again are club mosses, the pterophytes, which are ferns, whisk ferns, and horsetails, they do not produce seeds at all. But when we get into things like angiosperms and gymnosperms, they do produce seeds. All right, so this is kind of our progression. We've already seen green algae of various types, both the green algae, which are still green algae, as well as those that are more now classified with the plants and the Viridiplantae. We've seen our seedless non-vascular plants, so liverworts, mosses and hornworts, Hepaticophyta, Bryophyta, and Anthoceratophyta. Now we're kind of in this realm over here where we're going to deal with club mosses and ferns, and then we'll get into our gymnosperms and angiosperms. Now, the angiosperms, in addition to being flowering plants, are also plants that produce fruits. It doesn't matter what any other plant looks like, no matter what other structure contains on it, if it doesn't produce seeds, if it doesn't have flowers, it can't make fruits. To be a fruit, there has to have been the flower there first. Okay? So the advantage of fruits is that a lot of times fruits are attractive to things like animals, and animals can pick up those fruits, they can consume them, or sometimes the fruits can kind of hitch a ride on their fur or on their feathers, as we'll see a bit later on as well. But whatever the case, fruits allow for seeds to be dispersed by animals or sometimes for seeds to be dispersed by wind or even water in some cases. So fruits are a dispersal mechanism for plants, whether it's by animal or some other abiotic factor. Fruits are a very good way for seeds to get dispersed to other parts of the environment. So we're looking now at our Lycophyta. So this is our fourth phylum of plants. We've done Anthoceratophyta, Bryophyta, Hepaticophyta. Now we have Lycophyta. Lycophyta are the club mosses, and they are especially common in the tropical environments, but they do make it even in relatively far north latitudes. When I was a graduate student up in the UP of Michigan, very, very, relatively cold place, there are plenty of lycophytes growing up there. They call them princess pine or ground pine because they look like little tiny miniature pine trees, even though they're not even close to being pine trees. They look like it, so that's what people call them. There are a few species even that far north, that far away from the equator, but the closer you get to the equator, the more diversity you find of these club moss species. Again, they don't produce seeds, they don't have roots, they have some very, very primitive leaves, and they have those vascular stems as well. So, again, superficially, they kind of look like a certain kind of a moss, perhaps, but because this group has a sporophyte, this whole picture up here is a sporophyte that we see up here. Because the sporophyte's a dominant feature, we know these are not all that closely related to the bryophytes, because bryophytes, of course, have a more dominant gametophyte than sporophyte. The Pterophyta, are going to be the ferns and their allies. Allies are just related to them. That's kind of the name they use in taxonomy, is allies, related species. A lot of the ferns, especially the true ferns, are still in kind of a period of time when their taxonomy is still a little bit not known completely yet. So there are some true ferns that are classified as being more closely related to whisk ferns. There are some true ferns that are currently classified as being more related to horsetails. So it's still kind of in the process of being sorted out. So even though people have been working on taxonomy of these things for really a couple hundred years at least, based on new evidence that pops up, based on molecular evidence versus morphological evidence, a lot of this is still kind of in flux as far as how the classification actually works out. In this case, the Pterophyta, we're going to look at a fern life cycle. It's kind of our poster child for the seedless vascular plants. They form antheridia and archegonia, just like mosses do, just like just like liverworts do, just like mosses do, just like hornworts do. But their antheridia and archegonia are going to be quite a bit smaller in the grand scheme of things compared to the rest of the body of the plant, as we'll see. The pterophytes, all of them, whisk ferns, horsetails, and true ferns, are still a little bit reliant on water for reproduction because their sperm are flagellated, just like in the bryophytes, they're flagellated sperm, and so therefore they need water for dispersal and water for fertilization to occur. I always kind of equate the ferns and their allies to being kind of like the amphibians in that, when you think about mosses and so forth, they are very closely tied to humid or watery kind of environments, wet environments. Ferns, not so much, but they still have to have water for reproduction to occur because, like I said, they have those flagellated sperm, and without water, those sperm are not able to swim. So you're never really going to find a fern growing in a super, super dry place that never sees water. It's just not really a good place for them to be because their sperm wouldn't have a way to disperse and actually fertilize eggs. Whisk ferns are a tropical group. Really about the only time we find whisk ferns in this part of the world is if you happen to go into a greenhouse where whisk ferns can be a very, very common and problematic pest species. The thing with whisk ferns that's kind of unique is that they have what's referred to as dichotomous branching of stems, where if we let's see if we find one here, if we follow this stem up here, we have a stem, we follow it up, and it splits in two. And then that stem splits in two, and then that stem splits in two, and then they split in two, and they split in two, and they split in two. So it's called dichotomous branching. Dichotomous meaning every time it splits into two forks as a result, which is a pretty common strategy for these organisms to produce. Now, in this picture, something I want to draw your attention to, so we kind of go back and orient ourselves relative to sporophyte, gametophyte, sporangia, and so forth. These in this picture are all sporophytes. So the sporophyte is definitely the biggest and most dominant form. Gametophytes are very, very tiny and even microscopic in some cases in the whisk ferns, as well as true ferns and horsetails. So these are all sporophytes in this picture because they are large and they are conspicuous. These yellow structures on the stems are sporangia. All right, so if we think back to our life cycle about how ferns or how plants in general get through their lives, remember sporophytes contain sporangia. And it's within these sporangia that meiosis occurs to produce haploid spores. So when the haploid spores leave these sporangia, they go out into the environment by the bazillions. A few of them might find a suitable place to land, and when they grow, when they germinate, those spores will then form the next generation of gametophytes, which, as I said a couple minutes ago, are going to be very, very small, inconspicuous, microscopic in some cases, but the gametophytes do start the next generation of gamete-producing structures, which would then in turn allow for fertilization and the production of the next generation of diploid structures, right? Horsetails are a very, very small group of pterophytes. There are only about 15 species, and you can find these growing around here. Not a problem. They're common in the forest, depending on what kind of habitat it is. You can find them growing along roads because they're very resistant to salt and disturbance and those kinds of things. They don't need a lot of soil. They do really well in gravel, for example. Horsetails are unique in a couple of ways. They're all the same species. I'm sorry, all the same genus. There are 15 species of the genus Equisetum. So one genus with about 15 species in that genus is really about all there is in the entire world for horsetails. The sporophyte stems in this case come in a couple of different forms, right? So there are what are called vegetative stems, which is what this green stem is here. So vegetative stems are for growth and for photosynthesis only. There's no reproduction that occurs on a vegetative stem. There are also reproductive stems that have on the ends of them clusters of what are called strobili, and the strobilus then is a group of those sporangia. So again, each one of these little dots here consists of a sporangium in which meiosis occurs to produce spores, and when those spores go off to the environment, they can then land and germinate and produce the next generation of gametophytes. Right? So both of these two stems are sporophyte stems, even though they have different functions and different forms, right? Both of them have kind of this ribbed sort of nature where you've got a section and then a kind of a joint and a section and a joint over here, section, joint, section, joint, section, joint, and so on. Pretty common in horsetails to have that kind of arrangement. These also have a lot of silica in the stems. So silica, remember, we talked about silica all the way back as in the animal chapters when we talked about silica-based spicules in sponges. Silica is glass, essentially. And so within the stems of these horsetails, there are microscopic, very, very tiny, fine particles of essentially glass, which gives these stems not only structural support, but also a whole lot of protection against herbivory. Because if you're an animal that tries to feed on something that contains a lot of silica, the silica powder essentially in these stems is actually harder than your tooth enamel is. And so if you feed on them for any amount of time at all, they will actually grind your teeth down. And as an herbivore, that is definitely not something you want to see. So these plants basically enjoy more or less complete protection, at least from really big herbivores. There is almost nothing that will feed on horsetails because it will grind down their teeth. As kind of a side note, horsetails are also sometimes called scouring rushes because they have the sort of historical significance of back in the it's like pioneer day kind of things. As people were wagon trains going out west, that kind of a thing. They would take these horsetails and they would use them as almost like a Brillo pad to like clean pots and pans and those kinds of things because they're abrasive. They could actually use them to as like an abrasive material for cleaning, essentially. So they were called scouring rushes for that reason. Now the ferns are the biggest group of pterophytes. They're about 11,000 species, which is not bad at all for a big group of plants to have that many. But modern day ferns are nowhere near as abundant as ferns used to be back, especially around 300 to 400 million years ago when we were in what's called the Carboniferous period of life on Earth. So the Carboniferous was a time when this was like the peak of plant growth in the entire world because basically during this time, every single continent was either tropical or subtropical. So plants were able to grow like crazy back during this time. Now ferns back then could achieve heights of 30 or 40 or 50 feet. All right, so think about a fern like you would see growing in a house and make that thing 50 feet tall. Right, that's kind of the sorts of ferns we see. This is actually in this picture a tree fern, which these things can still grow. There's a few places in South America. There's a few places, I think, in Australia that has these. They get up to maybe 30 or 40 feet at most. But this is essentially a giant fern. Each one of these, what looks like a cluster of leaves, is actually a fern frond. And this thing is just an absolutely enormous version of what you would see growing in a house. Right? Now, the thing that's kind of important also back then Carboniferous period was that this plant productivity was so high and so fast that when plants died, they essentially died and built up on the forest floor in such great depths or thickness that decomposition couldn't break them down fast enough before another fern would like fall on top of them. And so you would get layer after layer after layer of dead plant material which couldn't decompose fast enough, so it built up under enormous pressure and eventually enormous temperature, and that actually gave rise to a lot of the coal deposits that we still have today that we take advantage of for our own use when it comes to making electricity. The vast majority of the coal that we still have today was developed back around 300 million years ago, and most of it actually came from large amounts of fern biomass, as opposed to other kinds of plants or other kinds of animals. So what we're going to do is we're going to wrap up this section now by looking at the typical fern life cycle. The life cycle here is quite a bit different than we saw before when we covered the bryophytes, so we're going to make some comparisons here, look at some similarities, and also look at some differences. We'll go ahead and start off up here at the gametophyte. So this gametophyte up here is called a prothallium. The fern gametophyte is called a prothallium, sort of a vaguely heart-shaped structure. But these are microscopic, or close to microscopic. They can be a little bit bigger, but really, really hard to find unless you're really, really trying to find one. These can be very difficult to find because they're so small. On this prothallium, or on this fern gametophyte, there are antheridia, which are sperm-producing structures, and there are archegonia, which are egg-producing structures. So these sperm cells, which again are flagellated, when it rains or when there's water present, can swim to the egg, fertilize the egg, and at that point we now have a zygote nestled within this archegonia. So at this point we have a diploid structure that's surrounded by haploid tissue. Now, this diploid structure is going to start to grow out of the haploid structure to produce an embryo, eventually to produce a very, very tiny miniature version of a fern, still growing out of the gametophyte here. So we still see the rhizoids, we still see the root, we still see the prothallium, but now there's a diploid sporophyte growing out of the haploid gametophyte. Eventually this grows into a full-fledged fern, which has rhizomes, which we talked about in a previous chapter, are the underground stems, which plants can use for asexual reproduction and dispersal. So both of these ferns on this one rhizome are genetically the same thing, but this rhizome allows the fern to actually spread throughout the environment and produce more fronds of itself. On the sporophytes, during certain times of the year, typically on the underside, there are clusters of sporangia called sori. So a single cluster is a sorus. Multiple of those are called sori. And a sorus is just a cluster of sporangia, again, in which we find meiosis occurring to produce spores, which are then haploid and can go off into the environment to germinate and produce the next generation of gametophyte, which can then restart sexual reproduction. All right, so that's a pretty typical pterophyte life cycle. A couple of other little features here you see down on this picture. These little guys that are starting to sprout from the rhizome, those are called fiddleheads, and those, a lot of times you'll see those sprouting up in the springtime. Right around the same time leaves are popping out on the bigger trees, you'll see fiddleheads growing on the forest floor, which will generate this next sort of generation of ferns for the next growing season. Usually, early spring, mid-spring, those are kind of the same time when you find fiddleheads first kind of unfurling themselves from the from the soil and giving rise to the next generation of ferns. All right, so this is basically what we just talked about on the previous slide. We've got sori, which are clusters of sporangia in which meiosis occurs to make spores. Spores are then haploid, and they create the next haploid generation of gametophytes. Again, in this group, still rhizoids, still don't have true roots, but now we have truly vascularized stems and very vascularized leaves as well. So the only thing these plants are lacking when it comes to vascular tissue is going to be in the roots. So from there, we're going to wrap up this section, and we're going to start up next with the gymnosperms, talk about some probably more familiar plants there, and then eventually we'll get ourselves into the angiosperms